Okay, so this is going to be the last video about energy generation, and then we'll switch to something completely different. So energy generation is a complicated thing that a lot of people like to skip over when they're learning biology. And to understand the effect bacteria have on the world, you really do need to understand their energy metabolism, because it really drives what's happening in your gut. It drives what's happening in rotting everything. Everything really in the world sooner or later experiences microorganisms trying to use it for energy. So it's important, and there's a lot of it, but I want you to focus on certain things. So do you need to memorize the formulas and reactions for everything? No, I think I've told you what you need to memorize. For example, you would need to know oxygen, nitrate, and sulfate, and know how they are used. What are they used for? What is the purpose of an electron transport chain? Why do we need an electron acceptor? Why do we need electron donors? Those things. How do those things contribute to energy generation in microorganisms? Yeah, so that's the same thing. You really should be able to use words like anaerobic respiration and fermentation and explain to people what they mean. And the words like facultative anaerobe and microaerophile and aerotolerant anaerobe and obligate anaerobe, those are words you really should know, because energy generation underlies a lot of what bacteria and archaea do in the world, but oxygen usage we can use to predict what human pathogens are going to do. And we do know how to classify those in terms of their oxygen usage, so you have to know the oxygen usage terms very well. So you'll need to be able to apply those terms and describe organisms with them. The last thing I'll say is I want you to be ready to talk about what happens in microorganisms without mentioning what happens in humans. So if I ask you about fermentation, ideally your answer would not involve lactic acid, and it would not involve a muscle cell. So there's danger always when you're taking a class of confusing things from your past with what you're trying to learn. And so if you're thinking of human physiology, that can be very helpful for learning any other kind of physiology, but just make sure you don't give me examples of human physiology when I ask for an example of microbiology. I've also added a slide to the end of this set of slides to help a little more with narrowing down what you're going to study. And I'm putting this here in the slides because I'm explaining it to you, but it's at the end of the slides for people who aren't watching the videos. So when we're looking at the pathways, the big pathways, and let's see what I'm talking about. When we're looking at, where is it? This sort of thing. I'm not expecting you, just to reiterate, I'm not expecting you to memorize all the ATPs and ADPs. I'm expecting you to memorize where the carbon is and where the electrons are. And so those are colored here. So the electrons are on the red or pink electron carriers, and the carbon is in the blue carbon molecules. I mean, all these molecules have carbon, but the blue ones are where the carbon from glucose goes. So pay especial attention to those colored molecules as you go through the slides and as you're studying. And when you memorize these, put them in terms of the whole pathway. The idea is make sure they all add up. Make sure we don't lose track of any NAD or NADH, and make sure that the number of carbons here, or as carbon dioxide, equals the number of carbons that were in glucose, that sort of thing. If you can see the whole pathway you can kind of see how it fits a little bit better, and any individual step is less intimidating. And then for slides that have electron transport on them, or that have representations of electron transport chains, there could be a lot of detail to study there. There are multiple enzyme complexes that translocate protons. They each have names. They each catalyze a specific chemical reaction. And there are specific electron-carrying molecules like the quinones and the cytochromes. But you don't have to memorize any of that. The only thing from an electron transport chain I'm going to ask you to memorize is ATP synthase. I will expect you to be able to explain why protons are being pumped across the membrane and what the different forms of energy are. And I do expect you to know how electrons get to the electron transport chain from something like glucose. But in terms of memorizing the actual electron carriers in the electron transport chain, don't worry about that. And that's this. Okay, so let's move on. There are other weird metabolic schemes. This is just a curiosity if you're nerdy. Yeah, I'm just going to skip that. And then something I've mentioned before is that fermentation products and some anaerobic respiration byproducts, they smell bad. And so anything that's lacking oxygen is going to smell bad. So why are these things lacking oxygen? That's a good question. Why is there no oxygen in human feces? Well, it's because there's so many bacteria there. The aerobes use up all the oxygen really quickly. So the oxygen is gone. If there's nitrate, organisms are going to use that up really, really quickly. So for the vast majority of time, there's no electron acceptors left over in the feces. And so fermentation is what has to happen. Also, like dental biofilms, same thing happens. Morning breath is an example of anaerobic bacteria smelling bad. Why do you think that would happen? You tell me. So I guess pause. Well, you keep your mouth closed while you're asleep. Most people do. And you're not adding oxygen or air to it. And ultimately, you can create anaerobic environments in your mouth as bacteria use up the oxygen and start fermenting, and you smell bad. So these are some oxygen-free environments. Like I've said, this is redundant, sorry. This is the hierarchy of electron acceptors. So this would be what someone like me could use to predict which electrons would be used in what order. So in general, oxygen is going to be the best, and an aerobic respirer is going to reduce oxygen, and the waste product will be water. Second best is nitrate, and they will reduce it to nitrite, and sometimes all the way to nitrogen gas. But this is half as good as oxygen. And then iron and manganese, these are often found in nature also, and these are not quite as good as nitrate. And then all of these you could just lump together. These are all kind of terrible electron acceptors, and the cells that use them for anaerobic respiration do not get much energy from it. But it is a source of energy. It's something in nature that can be exploited, so some microorganism will exploit it. So that is a thing. And fermentation fits in here kind of equal to these. So sulfate reduction would happen around the same time fermentation would happen. So while organisms have resorted to using these for anaerobic respiration, others would just do fermentation. But if you have any of these in the environment, and definitely if there's oxygen, there won't be any fermentation at the same time because the fermenting organisms can't compete. Right, and this slide just spells that out. And the organism that uses the best electron acceptor usually is going to win. It's going to be able to use up the other nutrients in the environment so that others can't use them. It consumes the available nitrogen and phosphorus. And right, so I've said all these things. So here's an example in human health. If a person has cystic fibrosis, this is a inheritable condition that affects their, well, many things, but also their ability to clear mucus from their airways. And so mucus will collect in alveoli and some parts of the airwaves. And that mucus is not sterile. And what you get, if you have, this would be the lumen of the airway, these are some epithelial cells, and this would be some mucus that is stuck in some area. Well, there are bacteria in it that will rapidly use the oxygen. So the oxygen is gone. Bacteria here on the edge get a constant supply of oxygen from the air, but they use it, and it doesn't get down here. So you have a layer of possible microaerophiles growing just under there, and then other things. Well, it turns out there's nitrate in this environment and some sulfate. And so what this does is this creates an environment where Pseudomonas aeruginosa can grow. And Pseudomonas aeruginosa is a gram-negative bacterium. It's kind of terrible. It's hard to treat. It is resistant to a lot of antibiotics. And it causes lifelong chronic infections in people who have cystic fibrosis. And yeah, it is very hard to treat, and I'll explain what biofilms are later. So how does Pseudomonas aeruginosa grow in that situation? Well, it's able to use the nitrate in anaerobic respiration. And this is important to it because it can't do fermentation. So if there wasn't an electron acceptor, it just couldn't grow at all. It's a very effective facultative anaerobe. So I think this video might not be the best video I've ever made. I think the slides are meant to really be self-explanatory. So if I confused you, just turn off the video, go back and look at the slides, and you'll be fine. So this is, again, the end of bacterial energetics and electrons and all that stuff, and we're going to look at two more topics in the upcoming videos. One is biofilms, and those are structures bacteria can form and live in, and they're everywhere in nature. They're on your teeth right now. And then quorum sensing, which is a way, it's a technique bacteria use to detect the presence of their buddies. And it's a really good example of how microorganisms see the world. So that will be in the upcoming videos. I will see you there.